Welcome to Industry and Identity, a career conversation with Dr. Yang Chai. Industry and Identity is a USC Asian Pacific Islander Faculty and Staff Association event series that invites USC API faculty, staff, and alumni to speak about their careers. In addition to sharing how their identities have influenced their journeys, guests will explore a variety of professional development topics. On behalf of USC APIFSA, thank you for joining us today. My name is Julie Shen. I'm one of the chairs of the Programming and Development Committee. For those of you who are not familiar with APIFSA, we are a relatively new organization founded in response to the increase in anti-API hatred and violence. Our co-chairs, Grace Ru and Linda Chu, formed APIFSA in April 2021 to create a safe space and a strong voice to advocate for API faculty and staff at USC. I am also the chair of the newly formed CAC APIFSA Inclusion Resource Group. The new IRG group was approved by CAC School of Medicine in December 2022. I would like to thank my vice chair, Dr. Lin Zhen, and the leaders of CAC APIFSA Employee Resources Group, Carrie Lee, Sue Lee, Cynthia Stander, and Trevor San Antonio for their help with this event. Our featured speaker today is Dr. Yang Chai. Dr. Chai is the university professor and the George and MaryLou Boone Chair in Craniofacial Biology at the University of Southern California. He serves as the director of the Center for Craniofacial Molecular Biology, CCMB, and associate dean of research at the Herman Ostrow School of Dentistry of USC. Dr. Chai earned a DMD degree from Peking University School of Stomatology, as well as DDS and a PhD in Craniofacial Biology from USC. He is most noted for his research on craniofacial development and birth defects, which has transformed the field and significantly improved our understanding of multiple craniofacial malfunctions. He also studies stem cells and is currently using innovative 3D printed scaffolds seeded with stem cells to regenerate tissue for patients who have lost bone due to trauma, genital defects, or disease. Dr. Chai is an elected member of the National Academy of Medicine and a member of the American Academy of Arts and Sciences. Dr. Chai has authored more than 170 scientific papers and numerous book chapters, and he recently edited a book on craniofacial development. Dr. Chai has been continuously funded by the National Institute of Health for more than 25 years. His work has earned him multiple awards, including the NIH MERIT Award and the 2011 International Association of Dental Research Distinguished Scientist Award. Without further ado, please welcome Dr. Yang Chai.
Thank you, Dr. Shen, for that very kind introduction. And thank you also, Shu and all the organizers for inviting me. And I'm really delighted to be here with you. So this is supposed to be a sort of a general talk to just share with you my scientific journey, how I got here, and then what I have been doing in my career. And my life is actually kind of pretty simple because I've only worked at two universities, at Peking University in Beijing, where I did my education and then also my clinical training, and I worked there as a resident and then taught there for a couple years before coming to USC as a graduate student. So just, this is meant to be a conversation. And if you have any questions, and you'll feel free to stop me. As Dr. Shen shared with you, I'm interested in craniofacial birth defects and stem cells in tissue regeneration. So our face is our identity. And we recognize each other largely based on our facial appearance. And we certainly appreciate the beauty of our the facial features. And when you have congenital birth defects, such as the one that you see on the screen, a patient with a cleft lip and cleft palate, that is very devastating. And not only for the patient, him or herself, but also for the parents as well. So this is how I started my journey, but I wanted to, go back a little bit further, tell you a little bit about where I grew up. So I was born in Beijing, and then I actually grew up right next to the Forbidden City. I always tell people when I was in high school, for those of you who can read Chinese, number 27 high school in Beijing, which is just right here, like next to Forbidden City. For our PE classes, we used to run around Forbidden City just, as a way to exercise. So, now it's harder to get to some of the areas, including Tiananmen Square. I joked with people, we used to sleep at night there, just, have fun. And things have changed a lot, but this was, many years ago when with my two daughters and my wife, we visited my old high school and, back to Beijing many, many times. So I graduated from the School of Stomatology at Peking University. And after that, I got into my training in oromaxillofacial surgery. That was at the time, I had an opportunity to repair these defects, such as cleft lip and cleft palate or other craniofacial birth defects. So in China, when we go through school called stomatology, it's kind of similar to a dental school, but it's much broader. It covers the entire craniofacial region. We do a lot of trauma surgery. We do a lot of head and neck surgery for patients with, head and neck cancer. And then my interest when I was in training was really on congenital birth defects because, at that time, and still even today, Asians have the highest incidence of cleft lip with or without cleft palate. And that's the second most common congenital birth defect right after cardiac malformation. And, it happens about 1 out of 700 live births, which is really, really high. For those of you, most of the birth defects are in the 1 out of thousands or tens of thousands. So from the very beginning, we were trained by some of the best surgeons in China at that time who, just done a remarkable job repairing these birth defects. But it was always very difficult for me to talk with the parents because they wanted to know why their kids were born with this particular birth defect. Some superstitious stuff or other people thought they did this and that. And it turns out, these birth defects can have a genetic component or an environmental component or a combination of both. But at that time, we had very little information. And every time I spoke with the parents, and it was just very difficult for me to understand, try to understand from their perspective and how they have to deal with this, and then also if they have another kid, whether this type of birth defect will happen again. So that was really the main motivation for me to seek out more advanced training. At that time, USC, USC has one of the longest running craniofacial biology program, just teaching about the development and birth defects in the craniofacial region. So that's how I decided to come to USC, and then did my PhD here, then came to this campus to do my postdoc, and went back to get my clinical degree from USC. So today, I spend most of my time doing research and teaching. I'm the course director for our head and neck anatomy class. I've been teaching with Dr. Snow, who's a faculty here at Keck for many, many years. And also I spend one day a week seeing patients at our faculty practice. I always really enjoy being able to interact with patients, and I do a lot of dentistry now and some oral surgery. But this patient interaction has always been the motivation for me to design my research projects. And then as you will see, I'll give you 2 examples. The cleft palate study is one of the longest running project that we have in our lab. I have a R01 that has been continually funded for 23 years. So that's the same number that has been renewed, over and over again. And that's, I'm very proud of that work. And then also you will see how this has really helped us to understand the disease mechanism. The other example I will share with you is a condition called craniosynostosis. So there are 22 bones in the human skull, with the exception of our temporomandibular joint, which is your jaw joint, as you are sitting here eating, drinking, and chewing. So you are exercising your jaw joint. So that's moving, right? All the other bones, 22. So you have 2 bones connecting together with the articulating joint, like your elbow or your knee joint. All other bones are connected by a fibrous tissue called the sutures. When these sutures fuse prematurely, as the brain growth happens after birth, that continue to push the skull to expand. But because the sutures fuse, so limit the skull growth, and kids end up with this very abnormal skull shape. This is not only a cosmetic issue. Patients with severe type of craniosynostosis also suffer learning disability and neurocognitive dysfunction. And what we have done is we're currently, I'll share with you some of the work my collaborator, the craniofacial surgeons at CHLA, what they're doing, and then what we're trying to do to improve the treatment of this devastating disease. So we'll start with a cleft palate. And so the cleft lip with or without cleft palate happens 1 in 700 live births. And then in this country, when a kid is born with a cleft lip and cleft palate, and normally they are repaired within the first year of life. Usually if it's a cleft lip, only within the first 6 months. With a cleft palate also may occur a little bit later, but as you can see, the surgeons really do quite a remarkable job and then to repair this defect. But what I hope you will also appreciate is this is not the only surgery these kids will go through. So from birth until 21 years of age, normally a child with cleft lip and cleft palate, they undergo between 10 to 12 surgeries. So first you do the repair, and later on you do a bone graft to get the alveolar ridge in shape, and then you can, because usually when they have a cleft, they have missing teeth, you have to put implants in, and then you have to do nasoplasty, try to reshape the nose, and then just to have a normal appearance. Same thing with cleft palate repair. It's also, just multiple surgeries coupled with speech therapy, and then a lot of psychological counseling and this and that. So it's a very long road for patients. Even they have the best care available in this country. So from many years of work, more than 30 years of studies, we have learned that cleft lip or cleft palate or cleft lip with cleft palate so clearly have a genetic component, meaning, here you see a father with a abnormal palate, and then here the daughter also has a cleft palate. So there are many cases that people have done these multi-generation studies. So, I don't have to tell you because all of us know the Human Genome Project completed in 2001. Based on this, we have done a lot of GWAS studies and then whole genome sequencing. All of this have helped us to understand what could be the link, between these birth defect and then some genetic mutation. And, we're in a cancer center, people all know BRCA1, the mutation was discovered many years ago. And then, celebrities like Angelina Jolie, who elected to have this double mastectomy, basically to prevent from cancer development, based on the information from, the Human Genome Project that discovered all the, or basically have the information of these gene mutations that can cause a certain type of cancer. So what we have done is to take the patient study based on the gene linkage analysis. And what we've learned is that TGF-beta mutation can cause cleft palate. And then, in patient study, basically what you have done, or you can see these case studies, is that you have a certain number of patients and they all carry the same mutation. So that's basically an association study. So you now know that this mutation perhaps is possible cause for this particular type of birth defect. Fortunately, we have the ability to generate these transgenic animal models in which we can mutate the same gene and then see whether that causes some kind of malformation. In this case, we mutated the TGF-beta receptor, and then that's the protein that helped to receive the TGF-beta signal. And so once we mutated this TGF-beta receptor, and then 100% of these newborn mice develop a cleft palate. So that really suggests, this is a possible cause, right? Now, aided with this animal model, you can study, what the mutation, how does that affect the palatal shelf growth, how the cells behave in the absence of this TGF-beta receptor. And then you can also look at what are the possible downstream events that have been perturbed, so that may be responsible for causing this birth defect. So one of the candidate gene that we discovered from the study was, so in the absence of the TGF-beta type 2 receptor, and then you have an elevated member of the TGF-beta ligand family, TGF-beta 2 was elevated in this mutant model. And then by generating a compound mutant, meaning that in the TGF-beta receptor mutant, you further reduce the ligand expression. and then you can actually achieve a 100% rescue, meaning that a palate will develop absolutely normally in these mutant models. So you probably there wondering, if you don't have the receptor, how does the TGF-beta actually signal, right? And we have actually done that study, and it was published a few years ago that shows that TGF-beta, in the absence of a type 2 receptor, can utilize some other receptors to initiate a sort of a ectopic pathway, normally not used. And that's supposed to be, that has proven to be detrimental for palate development. So now, these are the things where have been adapted into sort of a clinical practice to use this information for screening and also prenatal counseling. And then in the future, hopefully some of these can be utilized for some kind of intervention that will help to prevent or reverse some of this birth defects. So next, I will talk a little bit about craniosynostosis. And as I mentioned earlier, this is a disease due to the skull, the suture fusion. So on the left, on your left, yes, you can see this sagittal suture and the coronal suture. And then when all of these sutures are in place, and then the brain grows and expands, pushes the skull, and the skull will grow with it. And this sutures also, they're very important for, actually, we recently learned that this actually is a site where the stem cells are. And then without those stem cells, the suture will fuse prematurely. But here you can see this suture fused. So basically, you have a sagittal suture fusion. And then what you see, the shape, the abnormal shape of the skull, is basically the result of brain growth. So as you can imagine, now this is a fused side. You can no longer push the skull growth laterally anymore. As a result of that, the brain pushes the skull to grow anteriorly posteriorly. So you end up with this abnormal shape of the skull. Kids who are born with craniosynostosis, they will undergo surgery also within the first year of life, and that's most of the case. In some other cases, the surgeons argue that maybe you do it surgery later, and then the kids can tolerate the surgical procedure better. The way the surgery is done is basically the surgeons will go in and then remove this fused suture region along with some bone, and then they leave a gap in between. And then the gap or the two adjacent pieces of skull bone are secured by using this titanium plate. So this is really based on an educated guess. Sometimes you leave the space that proven to be too small, the bone can fuse again. So that will require a second surgery. Sometimes the gap is too big and the patient will always walk around with a gap in their skull. So that's normally the kids have to go through, they wear a helmet after the surgery in order to protect the brain because the skull bone is not quite covering the whole brain surface, right? So what we have learned from our study is that within these cranial sutures, these blue markers represent the stem cells. And they are, you don't have to worry about how we recognize them, but we have a genetic markers that we can actually use. And if you activate these markers that are blue and you wait for months, you can see these cells have crawled out, basically help to the skull bone growth. So that suggests you really need these cells to help the skull bone to continue to grow, right? And the way we test that, we ask that question is, what if we eliminate these cells? We just get rid of them. And we can do that in the animal model by genetically, ablate these cells. And then once we have done that, after 2 months, every single suture in the skull fused. So without these cells, basically you lose all the sutures. And then later on, we actually have reported, this was published in 2015. And if you wanted to see the publication, just email me. I'll be happy to share with you. In the a mutant model that mimics basically this gene was mutated in patients called the TWIS1 gene that causes craniosynostosis. We also have an animal model that exactly phenocopy the patient phenotype. And then in this animal model, you can see basically they lose these stem cells before the suture fuses. So that really provided sort of the inspiration for our next study. So we wanted to see how can we use these stem cells to regenerate a suture and then to build a new biological suture instead of cutting off a piece of bone, leave a gap, right? So this study was published in 2021, and then you can actually take a look later. So what we have done is to take advantage of these stem cells that we can harvest from a donor that's normal, and then combine it with this biodegradable material. And then afterwards, you can see we were able to use CT to show that we can regenerate a normal suture. If you don't put anything in place, the bone will fuse again. And then, or if you just put the material without the cells, then the bone will also fuse. But following that, the exciting part was, if you look at these wireframes, by regenerating a suture, we were also able to restore the skull size somewhat back to normal, much more improved than the red wireframe, which represents the TWIS1 mutant model. So not only, in this study, we were able to show that we were able to use the stem cells to regenerate the suture and restore the skull shape, but also restore the brain size and also improve the learning outcome of these animal models. And that really this has been really been a very exciting study. So now, with this in place, we're moving from the small animal model to the large animal model. And then as I will share with you, one of our study on the skull bone regeneration using the stem cells now has filed with FDA. We're in the process of actually getting the approval so we can conduct our phase 1 clinical trial. So far I've told you 2 stories about the 2 kind of individual R1 type of research projects. And next, I wanted to share with you just the 2 large consortium that we have been able to help to build. And one is related to dental oral craniofacial tissue and organ regeneration. And that's supported by NIH. And the other one is called a FaceBase, which is a collaboration with our colleagues from computer sciences, Carl Kesselman's group. And then we have built this data center where now NIH, the NIDCR, their funded projects are all directed to us to share their data related to craniofacial development so that can serve the broader community. So these are the 2 sort of a larger sort of a consortium we have built to serve the community. And then USC is leading these. Okay. So C-Doctor was built in, I think we started this program in 2017. And NIH was really very clever in a way to design this program. So the premises for NIH to start this program was that, there are so many exciting, innovative discoveries in that are happening in the research lab, but very few of these discoveries are making their way into clinical practice. So NIH wanted to fund this large program and then to help to bridge this gap from discovery to clinical implementation. The way they designed this was very clever. They built the program in 3 stages. Stage 1 was basically just build a team. So we applied along with many others. There were more than 50 applications throughout the country. And then NIH selected 10 programs to build the team that must include basic scientists, clinicians, regulatory science expert, material science expert, people know how to interact with FDA, people know how to interact with industry. So we started that, and then we quickly realized USC alone cannot do all of this, right? Because we don't have all the expertise. So after the first year, 1 year of planning, they said, okay, now you can apply for second stage funding. And then NIH decided to give out $20 million for each center that's funded. And then as a result, if you build a successful resource center, you get to solicit applications and then also give out these grant money so that you can help the development of these projects towards clinical translation. So we teamed up with UCSF, UC Berkeley, Stanford, UCLA, and then UC Davis and the City of Hope. We built this called a Center for Dental Oral Craniofacial Tissue and Organ Regeneration, abbreviated as CDOCTOR. And then we brought in local partners like USC Stevens, the QB3 at UCSF, and all the biodesign, CERSI. These are, when you talk about translation, you talk to the people at UCSF or Stanford, they do that as, like a routine practice. They have so many experience. So we really learned a lot. And we also have partnered with CERM. And also, of course, we continue to work with NIH people. And then, so we select these ITPs. And then before we did that, we actually went out and surveyed clinicians, asked them, if we regenerate any product for dental oral craniofacial, what do you think you can use on patients? And then we started a program based on all of that. And then it's really informed by clinical practice. And then we build these resource center and the service. And now we have 3 projects within the last 5 years that have filed with FDA, IND filing. So that's a pretty short runway, right? So you think about that. So the focus is really about how we can regenerate tissue to have a better repair for cleft lip and cleft palate. How can you deal with patients who have large part of missing skull bone? Right now you just cut other bone tissue and then try to patch it. But how do we repair these large traumatic defects? So that's really what the program wanted to do to enhance a clinical translation. And then these are the ITP, the projects that are being supported. As you can see, it's about reconstruction of irradiated soft tissue defects, prevention of scar formation in the skin. We have a calvario bone regeneration project, a stem cell-based therapy for bone regeneration, regenerating salivary glands, craniofacial muscle regeneration, and also repair for cleft lip and cleft palate. So each one of these projects up to now have probably received between $1.2 to $1.8 million to support this sort of a preclinical development and in order to get to the FDA filing. The second part, is about this face-based, which is built as a comprehensive resource to a data center to support craniofacial dental research and clinical research. And then people will access data here, but they feel confident these data are well curated and then can be shared and then can be cited and then so satisfy the NIH data sharing requirement, but also now, we have accumulated a lot of these 3D facial scans coupled with genomic data. And then now we have done a lot of these studies using AI and machine learning, see whether those information can be used to predict some type of a birth defect. Oh, here. FaceBase really serves as a one-stop shop for craniofacial researchers.
Although about half of all birth defects involve the face and skull, scientists remain unclear about why most occur. A comprehensive and systematic understanding is needed of how the faces of healthy children develop and what goes awry to cause common malformations. With today's improved technologies, researchers can generate many gigabytes of information in a single experiment on hundreds of genes that are involved in the process. FaceBase was created to integrate this information into a meaningful whole, while adding new data to form a more comprehensive picture of this complex developmental process.
Research in craniofacial dysmorphia could be accelerated and harder problems solved if researchers were able to collect and share their data and make it easier to find data that other researchers had produced.
In the past, research data was either not shared or organized poorly, with thumb drives lost in the abyss of filing boxes by graduate researchers long gone before publication. Some journals have ad hoc repositories, but are neither standardized nor connected with other studies. FaceBase is a comprehensive data repository that systematizes how data is presented. Basic curation and an underlying platform allow intuitive data entry with a wealth of metadata that add context and connections with other studies. FaceBase has become the established industry standard, adhering to fair principles for best practices in sharing and reuse of data for the craniofacial research community. Findable, accessible, interoperable, reusable. FaceBase datasets are validated and curated before release. Most datasets have multiple replicates to ensure data quality and reproducibility. FaceBase serves as a springboard for hypothesis-driven research, providing access to data far beyond what any one lab could generate by itself.
By sharing data at FaceBase, you have contributed to a platform where we can cross-reference each other's data and validate each other's data. You can use this platform at FaceBase as an opportunity to start future collaboration by working with others that will enable us to make discoveries that we could not do by working alone.
Submitting to FaceBase helps satisfy data sharing requirements from funding agencies like the NIH and from journals. Improve the reproducibility of your results by sharing well-documented, reusable data. Increase the dissemination and impact of your research with full attribution, DOIs, digital object identifiers, data citation guidelines, and citation metrics.
So we've invested a lot of effort in understanding the sociology of the research community and the craniofacial researchers, and ensuring that control and dissemination and credit of the data is done in ways that will make them comfortable.
Over the years, we at FaceBase have developed a standard pipeline for data submission. We have streamlined the process and then developed training tools and also very standard protocol how to submit their data to FaceBase.
As researchers continue submitting data, FaceBase will continue to grow and become even more invaluable to the research community.
The technology that we're creating and advancing in FaceBase is being used and informs research in many other domains. And so what we learned from FaceBase has broad application to the advancement of biomedical science in general.
Someday in the future, that we will be able to connect all these data resources for people who work in any parts of the human body, and then make them into one comprehensive and also ever-growing data atlas.
FaceBase is really an exemplar of how science should be done in the future.
So, the reason I share this with you is because not only we really are motivated with our research interests, but also we help to build these larger consortium, so can really help to serve the community, so that both clinicians and basic scientists can continue to benefit from all what has been generated. So I've really been very fortunate to get recognized for this, but I think I really credit all of that to my family, and the support from my family just has been unparalleled, and really couldn't have done this without their unconditional support. So with that, I'm happy to take any questions, and thank you very much for sharing this time with me. Thank you.
Dr. Chai, thank you so much for your presentation. I really appreciate your work. You have done a lot of research. I also appreciate the end you mentioned the face-based. I think that shared database is really important. Like I'm in cancer research, and we also have a similar database like the TCIA, like we have the cancer imaging archive, like people can share. So I think my question for you is related to API identity. So I think I share a very similar journey. I am also from mainland China, and then I came to the US for education, and then I stayed. So during your career journey, have you encounter any obstacles related to your API identity? And then on the other side, do you think any of your Asian API characteristic actually help you succeed? So I guess it's two questions.
Yeah, thank you for that. A very important question, really. I think we're here because we have been really fortunate to have a lot of great mentors and colleagues who have really supported us over the years. For me, I credit a lot to my mentors back in Peking University, the surgeons who really taught me a lot of these, how to really interact with patients, how to be a good doctor, right? And then, and from there, just working with many very influential people. And I always feel we're fortunate to be in an academic environment where whenever we have a question, doesn't matter if it's related to research or related to what you should do in your personal life, you can always find someone to ask questions and then get advice and then help you to make a very informed decision. In terms of being, Asian, or I've always been very proud to be someone from, grew up in China and then came to this country. I think, the United States has provided such a wonderful environment to support the career development if people are willing to work hard and then try to make a difference. And, I think we also want to continue to help the next generation to be able to advance their career. It is not always easy. We see always now, especially more recent years, see a lot of these negative news and related to people, being targeted. And I think we really need to work together as a group to support each other, not only just Asians, but also, as immigrants to this country, right? How do we really help work hard to contribute the advancement of science or technology or anything you do to really make our own, to make the Chinese proud, to make everyone proud. And so I think it's a ongoing effort. As a part of a, with an Asian background, I really haven't felt anything. Most of the time, I just feel people are very supportive and then being able to accomplish what you need to do. I can't say I have been, I've been very fortunate. I think, it's very unique, and maybe others have some experience that we can, use this platform to share, and then we can have a further discussion. But for myself, really, everything has really worked out very well because, many great people, my postdoc mentor, my PhD mentor, and then my colleagues now, every time, I needed some advice, I've always been able to get. And my approach is always try to surround myself with smart people who do things differently than I do. Even in hiring junior faculty, we don't hire just craniofacial people. We hire people who work in neuroscience, people work on cardiac development, working on skeletal. So all, working together, that's how we can actually do things beyond the craniofacial. Because when you have defects here, but also affects other part of the body, right? And so that has been really a main driving force for us. Any other questions?
Thank you. Very impressive presentation. I'm Ling Zhen. I serve as a Chief of Clinical Analytics for Keck Hospital and Norris Cancer Hospital. So because of my profession, working a lot with data, I truly think the face-based is very impressive. So I think in your role as a leader, educator, and researcher, I kind of wanted to know a little bit more. How do you come to this idea of creating the face-based, and how do you collaborate with people to bring in all the structure or all the team member needed to get it going?
Yeah, so thank you for that question. So face-based was actually started by NIH. So they wanted initially the program was designed as a spoke and a hub project. So if you think about like a bicycle wheel, right? You have all these connection to the hub. And so the idea behind that was to focus on craniofacial birth defect. But then NIH issued this call for application, and then they selected a data center, but also they, through peer review process, they selected 10 research projects that supposed to feed data into the data center. At that time, the data center was actually at the University of Pittsburgh. And so make a long story short, we have some really fantastic data scientists at USC who actually know how to manage large data. And so this is, we started this journey more than 10 years ago. So when the second competition came up, that was after 5 years of initial funding for face-based, and USC, so we teamed up with my colleague Carl Kasselman, you saw on the video. So we went in and competed to be the hub, and then we were successful. So then the data hub came to USC, and then for the last 12 years, it has been, more than 12 years, have been at USC. For recently, the NIH made a decision. They decided instead of having only 10 spoke projects feeding into the hub, they are requiring all NIH funded research projects related to craniofacial development to deposit their data to face-based. And that couldn't come at a better time, because now we also are required by NIH to do data sharing, right? So face-based, just kind of has evolved because the not only, we have been able to run this hub, but also we've been able to demonstrate the success. Data can be uploaded to FaceBase. Let's say if you have a research data, then you can control. So you don't want to publish this right away until, let's say, your paper is published. And you can actually time that until the paper is published or accepted. You want to share, and then at that time, the data will be released to the public. Not only we do that, but also we link to many of these other data sources, like dbGaP and then Geo and all of this. So you can use this as a platform, and then to bring all these data sources together. And then now we're actually expanding more into clinical data to bring in more disease models, so we can have the clinicians come in, not only use this to search for some of the information they need, but also as a platform to talk about what's the best treatment option that they can do for their patients. So use that data science to support making an informed decision for future treatments. So I think, this has been growing, and there are other data sources actually also at USC that supported by Carl's group. There's called a good map that focuses on kidney and the urogenital system. They also had a lung map on lung development. There's a brain map on the brain MRI imaging. So that's why we wanted to link them all together eventually to support research. Question? Yes.
Thanks, Dr. Chai. My name is Walt Wan. I work at University Communications, so I just came from the University Park campus for this presentation. Thanks so much, and I was amazed to see all the slides. I'm obviously nowhere near the medical expert, but I was so amazed to see all of the materials that you just presented. So I'm also a APIFSA member. I actually together with Julie and Shu. So my question is simple. I'm just curious how, obviously there are tons of research directions in the medical science area. I'm just curious why you selected craniofacial biology as your focus and as your expertise area.
Yeah, so, thank you for that question. I mentioned earlier when I was doing my clinical training, I had a lot of these patients with craniofacial birth defects, such as cleft lip and cleft palate. And, I couldn't really address the questions from parents. And that was kind of a motivation for me to come to the US to study. And then, even today, all of our research focus areas are all really informed by clinical problems, like craniosynostosis. We also work on tooth development and then root malformations. But these are, just some of the challenging problems we face as clinicians broadly. And then, like a surgery when you have kids with craniosynostosis. I mean, you don't want to just, the surgeons do a great job. I mean, really, they separate the bone. But if you can get a biological suture that can continue to grow as a skull, wouldn't that be wonderful, right? You don't have to do a second surgery, and then it's a biological solution instead of a mechanical solution. So that's how we are kind of motivated and informed by the clinical needs to continue to work in this area. Oh, I think that was just a comment on, sharing research career journey with us. How did you build a diverse research group? And how did you encourage team members with different culture and educational background to work towards a common goal? Oh, so yeah, thank you for the question. I think, we probably can spend a whole day talking about this. This is how do you get very diverse people working together, and how do you build a culture to support the development? And it's very hard. I can't say other than that because it doesn't always work out. I always felt, whether you recruit graduate students or postdocs, you will wanted them to be successful, but you have sometimes to understand your definition of success may be different from what they want to be defined as success. I think it's very important to understand what is the goal of your trainees wanted to accomplish, and then provide our best opportunity for them to do so. And we do the same thing for our junior faculty. I think I can tell you this, even with our postdocs or junior faculty, they have been really successful in competing for faculty jobs or getting promoted, tenured. Once we get our junior faculty hired, we form a mentorship committee, and they meet every 6 months, and then to go over their goals, and then to make sure they hit their milestones. So, they all come from diverse backgrounds, but I think if you have this kind of culture that build this mentorship structure that will constantly set goals and review, and then be able to help them to accomplish what they need to accomplish. And most of the time, I think things will work out, and there are sometimes don't work out, and then I guess you have to make some difficult decisions. But yeah, that's I think in the short version, that's what I can say. But it's very challenging. I would say it's getting even harder now because of we really they're so hard to find postdocs now, good postdocs, and but we continue to try and to train people, and then yeah, it's not easy.
I have one more question, because I think for the industry, as an industry leader, actually I'm wondering, for the early career researcher or career seeker, what would be your just one most important advice for them? At the very beginning, developing career. I think we all know it's very difficult, but what do you think is the most important quality, or even character that help you in your journey?
Yeah, I tell my trainees and I tell my kids the same thing. Really follow your passion. There's everything I can teach, in the lab about doing research or clinical procedures or things like that. It's not really difficult, but I can't teach passion. If you don't like what you do, really, there's nothing I can do. Because, life is hard, right? People see the success, all the different papers you publish, this and that. I can tell you without any reservation, I get more rejections than anything else. Sometimes you send out a paper for review, they don't even review, they send it back, say, oh, send to a different journal. It's okay, but if you like it, you think this is really a impactful study, and you keep pushing, you keep trying, but you have to feel excited to get up every morning, say, I really want to get in to do my work. Otherwise, work itself is already very challenging, very demanding, and as you get older and you get more responsibilities, if you don't like what you do, it's really, really hard. So I think what is the beautiful thing about this country is you don't have to do things you don't like. You can always follow your passion. You can change your career. You can do different things, and then just really be just ask yourself, what are you excited about every day? And I always tell my kids, just really make a decision based on what you think you would like to do for the rest of your life. And I think that's very important. That becomes a major driving force for your career development. Yeah.
Okay, thank you so much, Dr. Yang Chai, for being our speaker today. And thank you to everyone came in person and tune in online. And if you are interested in joining USC APIFSA, you can check out our website, apifsa.usc.edu. You can email our chair, apifsa@usc.edu, and that's APIFSA@usc.edu. Last but not least, I would like to acknowledge our two sponsors, the Keck APIFSA IRG on the Keck School of Medicine side, and the Keck APIFSA ERG on the Keck Hospital side. And if you want to become a Keck APIFSA member, you can email keckapifsa@med.usc.edu. So there are two organizations. One is more on the UPC campus and one is on the Keck HSC campus. So May is API Heritage Month. So we have organized many events. So there's a haiku contest, photo contest, Asian snack cart, and brown bag lunch. So please stay tuned. Thank you so much for being here today. Hope to you'll come join our other events. Thank you.